Hello, everybody. It is a great pleasure to be part of this course. My name is Gregor Kasprian. I'm a radiologist from Vienna, and I love to talk a little bit about fetal MRI, and I would like to share some thoughts on when fetal MR makes a difference. Now, that's the place where I'm working right now in Vienna at the Medical University. For those of you who are still reached that age to be a fan of Starship Enterprise, I grew up with this TV series. You may remember that they had something like a biobed, which is a device where you just hit a button and you get all the clinical and physiological information of the patient just in a matter of a second. I can tell you that fetal MRI is often thought so in that way, but I can tell you that fetal MRI is not operating in that way. We need expert radiographers, people who are actually operating the MRI. We need an optimized scanner for fetal MR. You cannot use a scanner from scratch. You need to optimize your scanner. You need optimized sequences. You need to develop and optimize your given MR sequences to a state where you really can make them usable for fetal MRI. And then finally, you need expert radiologists. You don't need a radiologist who has seen a fetal MR once in his lifetime. You need to have experts with a lot of experience. These are the guidelines provided by ESOAG on fetal MR. You see that neurosonography is basically the starting point where we are actually start working on from. We can add significant information. We can also even replace in some areas where there is not advanced fetal neurosonography available, neurosonography, but this should change after this course. So if you really are into fetal neurosonography, MRI is your companion and should not replace you. However, you want to make sure that at the same time you're developing neurosonography, you also develop your MR imaging program. You need your radiologist on board. It's a really fun and very, very insightful examination, also for expert radiologists who want to develop in that area. And there are quality guidelines also for MRI. It's not only that ultrasound needs to have a certain quality, also MRI needs to live up to some standards. What I would like to do in the next 20 minutes is to talk a little bit about the properties of fetal MR and to talk about what are the differences between ultrasound and MRI and how can we use them to our advantage. I would like to integrate that into the concept of fetal MR phenotyping, and I'm going to talk about that in a minute. I would like to incorporate that in some sort of different pathologies. Of course, I'm unable to cover all the neurodevelopmental pathologies we can see and characterize on MRI within 20 minutes, but I picked some of those just to use them as examples where you can gather more and complementary information. I would like to draw in the beginning a comparison to astronomy. In astronomy, you can look up to the stars using a conventional telescope. By that, you can see the optical light and maybe you can see a galaxy. However, if you're really into astronomy, you would love to see many, many different other informations. You want to maybe try to see beyond the optical light using infrared or radio waves. You want to see the ultraviolet light these galaxies are basically emitting, or you want to do even X-rays. All of these different components of light are conveying a different sort of information. Here with optical light, you will have to see the morphology of the galaxy. If you know how much CO2 is contained within those stars, this will give you a good map using radio waves. You also can get an idea about the size of these stars by looking at the sort of light they are emitting, if it's either infrared or UV, or you can also look at the energy they are emitting, and also this is a very interesting information. And that's exactly how MRI operates. It just does not only give you the morphology, it also tries to give you information beyond just structural information. The structural information, and I'm convinced about that, is already covered by neurosonography to a really, really very advanced stage. Here is an example, a picture provided by Ritsuko Bu, really excellent 26 gestational weeks image, and these are the corresponding MRI images, and you can see the structural information given by neurosonography is already very, very informative. However, MRI is adding other sort of information. Beyond anatomy, it provides information about tissue microstructure, maybe metabolism using spectroscopy, connectivity using tractography, function, and this is a still evolving part of fetal MR by looking, for instance, resting state pole activation. And finally, we can also look at standardized growing brains, so-called atlases, which provide a normative information, and we can actually look how different a given case is from that normative information. Fetal MR, as said, is challenging. You often get images which are not excellent. You try to get images which are sagittal, coronal, and axial, and out of those you have to be aware that often we are seeing a lot of motion artifacts, and we can only focus on those with the most optimal quality, and that's actually those we pick for our evaluation. Meaning, this is an examination happening not only by hitting a button, it's very interactive. We are always checking what is the quality of images we are provided with. For instance, we have images like that within our data sets, which we have, of course, to exclude from our analysis because they are restricted by motion. Sometimes you see these really distorted brains where you think, oh, is this a craniosynostosis or what's going on here? In fact, this is a susceptibility artifact due to air, here in the case, in the rectum of the mother, which is distorting the magnetic field and causing this sort of artifacts. You have to be familiar with these artifacts in order to assess your fetal MR examination, and that's what I'm talking about. This needs expertise. We want to be perfect in the orthogonality of our planes. We want to be perfect in a perfect sagittal, coronal, and axial plane. In that axial plane, for instance, we check if the lenses are in one plane, but we also can look at the so-called 5 spot sign. These 5 spots are very informative because we see the optic nerves, the pituitary stalk, the infundibulum, and the internal carotid arteries on both sides. And actually, if you don't see those, there may be something wrong. If you look here on the axials, you see on one side the optic nerve is smaller than on the other, and on the coronal one also one side of the optic nerve being smaller than on the other side. This is just one example to show how important it is to hit the fetal brain in the perfect orthogonal plane. And that is a very, very important message in the ESOC guidelines: use orthogonal planes. Also, use other sequences such as a T1 weighted sequence and a T2 star sequence. The T2 star sequence is often underestimated in its diagnostic power and information, but it is very, very worthwhile by the information which is provided through this sequence. This is a case at 24 gestational weeks with a vein of Galen aneurysmal malformation. You see the dilated vein of Galen and the surrounding brain here. On the first glance, on MRI, T2 weighted sequences as well as on neurosonography, this brain looked normal. If you would run, in addition to your structural anatomical T2 weighted sequences, a T2 star weighted sequence, you suddenly notice these susceptibility artifacts, meaning that there are post-hemorrhagic remnants along the ventricular wall, meaning these could also well be dilated veins. We have some difficulties in discriminating those. However, this means the parenchyma of this brain is already suffering from the malformation. It is already providing us information that within the brain, despite of a normal structural anatomical scan, there is a pathology going on, which is crucial in the determination of the prognosis of this choroidal type of vein of Galen malformation. We can add a DWI sequence which further is confirmatory of this pathological process, showing diffusion restriction along the ventricular walls, furthermore indicating that there is a damage to fetal brain parenchyma. This is another case. We have a small transverse cerebellar diameter. This is not usually a typical sign for a Dandy-Walker malformation. You know, many of us are looking for the position of the torcular when we are talking about Dandy-Walker. This was normal here. However, we see a small vermis, which might be slightly upward rotated, but not really. But still, some people might call that Dandy-Walker, which to my view is wrong. It's even more wrong when you consider doing other sequences here and getting this other sequence information like T2 star here, where you see exactly at the side of the cerebellum hemosiderin deposits. So there has been a hemorrhage to the cerebellum, and this is not per se a Dandy-Walker malformation. This is the original examination at 21 weeks, where you clearly see this hemorrhage sticking to the cerebellum. You're wondering why this hemorrhage is not spilling out into the subarachnoid space. Well, we'll come to that in a minute, but before I would like to show you the follow-up of that case. This is Sophie at the age of 5 years, and she's doing well. So you can imagine, particularly in hemorrhages, the origin of the hemorrhage and the reason for the hemorrhage play a huge role. She is somewhat clumsy in her motor profile. However, cognitively, she's doing well. So what are we seeing here? We are seeing a so-called subpial cerebellar hemorrhages. Cerebellar hemorrhages are mostly happening to the granular layer, or even more superficial, to the most outer part of the cerebellar cortex. They are typically not spilling out into the ventricle or into the subarachnoid space. They are attached to the cerebellum, which you can clearly see here in this anatomical and pathological specimens. What is also striking in our analysis of these hemorrhages was that you can clearly see here the pia, the superficial border and membrane encircling and embracing the cerebellum, and just underneath it you see this reddish material here. That is the hemorrhage. If you do a certain immunohistochemistry staining, you will also realize that just at the side of these hemorrhages, the glial cells, this is a normal case with normal glial cells in brown, and this is a pathological specimen adjacent to the hemorrhage, where you see there's a loss of the so-called Bergman glia, meaning these hemorrhages are not only destroying the cerebellum, they seem also to introduce changes in the local glia cell production, and they are probably leading to local changes in the microstructure of those tissues. So there are things happening beyond our way to visualize those on ultrasound and even sometimes on MRI. So DWI is very informative, and the analysis of the fetal brain parenchyma is really one unique selling point of fetal MR. It provides not only this T2 weighted structure, which is more or less comparable to ultrasound, it also gives you information beyond these structures, looking into the different layers of the brain parenchyma. This is a very classical image I use a lot to just exemplify this. This is T2 post-mortem. You see the cortex is usually dark. Then we have the sublate, which is rather bright. And around the ventricles, the intermediate zone and ventricular zones. The ventricular zones are very cell-rich and they are very dark. This layering exactly can not only be seen on T2, but also on DWI. You see a cell-rich cortical plate, sublate, intermediate zone, and periventricular zones. And all these zones can also be seen by another sequence, which is sort of a trick. What are we doing here? We are introducing a signal to suppress water. And with this water suppression, we get rid of the extracellular matrix rich water here of the sublate, and by that the contrast within this brain parenchyma is even more accentuated. Why may we use that? This is an example of a cortical malformation, and if you do T2, you see the irregularities on the cortical surface. This is something you might see on ultrasound. In this case, it was missed. However, by decent neurosonography, you might identify these changes. What you probably have a hard time is to see beyond those within this fetal brain parenchyma to understand the interruption of the sublate here, which is not seen as in other parts of the brain. This is the pathological specimen showing also these cortical irregularities here. Now, I would like to move on from the rather methodological aspects of fetal MR to the aspect of phenotyping. We're living in the era of precision medicine. Precision medicine is basically telling us and suggesting us that we can tailor disease prevention and treatment by a very accurate analysis of each single individual. Of course, genetics plays a huge part in that. However, also MRI and ultrasound will provide data which allows us to provide a very comprehensive and complete analysis of what is going on. So phenotyping is the precise and comprehensive analysis of abnormalities. However, we already know that not all of the phenotypic features of a disorder are often covered. We might see some sort of malformation at some place. We might see callosal agenesis. However, we might miss some very peculiar aspects in the posterior fossa, in the hippocampus, in the fornices, in the layering of the brain. All of these elements, in my view, need to be assessed not in 20%, not in 40%, in 100%, in order to give a very precise prognosis. And here we rather have in the future, we have to think about images not only as images, and the discussion is not important who is doing these images. This is going to be less and less important because our systems are getting better and better, and these expertises can be partially taken over by computers. However, there will be a huge amount of data coming from either MRI or ultrasound, and we have to deal with this data. Network medicine is one way to analyze the data and to assess the complexity of this information which we are getting. The more information you get, the more tools you need to make them understandable. With network medicine, by these network graphs, we have a handy method here which allows to identify commonalities, but also differences between two different disorders. We believe that this sort of looking at data will become very important in the future. We use that, for instance, in cases of callosal agenesis. These were 100 cases of isolated callosal agenesis, and we looked at how frequently we can see abnormal lamination, how frequently we can see an abnormal hippocampus, or how frequently we see abnormal basal ganglia. And you see, these things can occur in callosal agenesis. However, some of those things are really comparing very, very frequently, whereas others are really very rare. By these sort of graphs, we try to understand which associations might harbor a rather unfortunate, a rather bad neurodevelopmental outcome, and which of these associations, together with genetic information, might be of a more beneficial outcome. We try to do that in our prognostication score in isolated callosal agenesis, which covers a series of different phenotypic elements: hippocampal position, hippocampal size, asymmetry pattern of the hemisphere, amount of colpocephaly. All these features go into the score. The more of those features are present, we think, the higher the risk for a more complicated postnatal course. And so far, this was, at least in our series, true. Another way to use network medicine is to combine different organ pathologies with each other. You know that heart and brain is not only the matter of this course here, they are strongly associated with each other through physiological processes. So it makes sense to look at the phenome of congenital heart disease cases. In this case, this was our cohort of over 400 cases, and look at which features MRI is basically identifying and see which associations are present there. And what we found here was very striking. We saw that there is frequently an abnormal placenta, there is frequently an abnormal and too small cerebellar vermis, and finally also ventriculomegaly was also very present in this network. So there seems to be an association between congenital heart disease and some maldevelopmental processes in the fetal brain. We can also go into more detail in the future, and I said images are data, and we actually can advance this data. We can make the data more brisk, more accurate by AI-supported image reconstructions. Here is a comparison of this image reconstruction before and after using AI, and you can see that at least on MRI, the cerebellar vermis is really seen in, I feel, unprecedented detail. This is important because we now know that within the cerebellum there are compartments which play a role. And one of those compartments is this structure right here. This is the rhombic lip. And the rhombic lip plays a huge role in the development of the entire vermis and the cerebellum. And sometimes this rhombic lip can be transformed into the so-called Vermean tail sign, which you can see right here. So the Vermean tail sign is a frequently encountered feature in so-called Dandy-Walker malformations, besides the flattening of this angle here, which is also very important to be assessed. This is the fastigium angle, and if it's flat, it's more of an indication that we are looking at a so-called Dandy-Walker phenotype. Now, this rhombic lip produces all the cells the vermis needs to grow, and so it's understandable if there is a pathology going on here, it also will affect the growth of the vermis. What I would like to share with these final slides with you is that in so-called Dandy-Walker phenotypes, there is often an absence of the rhombic lip histologically. The development is frequently delayed, and only in the smaller part of cases, the rhombic lip is really present and normal. This also goes hand in hand with knowledge from Kimberly Aldinger's group from 2019, that the minority of cases with Dandy-Walker will provide you a genetic diagnosis, meaning that the Dandy-Walker phenotype might be more frequently associated with epigenetic, maybe also hemodynamic factors, here's the link to congenital heart disease, than with genetic disorders. And this is for me a strong case that we have to be aware that currently still the minority of cerebral pathologies is genetic. The larger part is either unknown or has also an epigenetic acquired background. The only way currently to address this properly is by an accurate phenotyping examination. You can use ultrasound for that, but if you would like to see the entire spectrum of all properties, remember the galaxies I showed in the beginning? It's probably a wise thing to also use MRI. I hope that I could share some thoughts on using MRI. I hope that after this talk at least, everybody understands that MRI cannot just be done from scratch. You need experience, you need training, and if you want to build up a center also harboring and getting and collecting all this imaging data in order to provide a very, very deep phenotyping assessment, you really need to collaborate with your radiologists to build up that sort of service. And with that, I thank you for your attention. It was a pleasure again, and I hope you have an excellent learning experience during that course, which is really widening your mind in the arena of fetal imaging. Thanks a lot.